Morning, good afternoon, or good evening. Can you see the slides?
Yes, yes.
Okay. First of all, I want to thank the Scholar Conferences for inviting me to this prestigious virtual meet. It is an honor for me to be able to share my experience with you. Today, I'm going to talk about our recently published work entitled Isolation, an initial characterization of human glioblastoma cells resistant to photodynamic therapy. The other authors are from Universidad Nacional de Rio Cuarto and Universidad Nacional de Córdoba, Argentina. Glioblastoma is the most malignant type of brain cancer. Although several treatment options are available, including surgery, chemo, and radiotherapy, the disease has a very poor prognosis. Patients have a life expectancy of around 14-15 months. It was observed that the surgical resection guided by photosensitizer fluorescence followed by photodynamic therapy or PDT prolongs the average survival of the patients. However, one of the main problems with all oncological treatments, including PDT, is the presence of resistant cells that cause the disease to reappear. One of the drugs used in PDT is aminolevulinic acid, or ALA. ALA is converted into the cells in the photosensitizer called protoporphyrin 9 through the heme synthesis pathway. Cancer cells have enzymatic failures that favor the accumulation of intermediate metabolites, mainly protoporphyrin 9. ALA derivatives, such as the methylated derivative methyl ALA, has been synthesized, which aim to improve the incorporation into the cells. But so far, they are not used for gliomas. The property of fluorescence of the photosensitizer is used for photodiagnosis and for fluorescence-guided resection. First, ALA is administered to the patient. Then the surgeon removes the tumor aided by the fluorescence of the photosensitizer. And finally, remaining cells are irradiated to cause their death, but reactive oxygen species. But unfortunately, some cells always resist treatment, generating recurrence in a few months. Studies on glioblastoma resistance to PDT could be of value in improving treatment outcomes. So the objective of our work was to isolate and perform an initial characterization of human glioblastoma cells resistant to PDT, employing methyl aminolevulinic acid, or methyl ALA. Our PDT resistance model consisted of applying several rounds of PDT to glioblastoma cells of the human cell line called T9AG. Cells that never received treatment were called parental cells, and cells that received 7 cycles of PDT were called resistant cells. By an entity assay, we saw that resistant cells in red had a high degree of resistance comparing with parental cells. And we wondered why the cells resist treatment. One of the causes could be that they accumulate less photosensitizer. As we can see in the photographs, the fluorescence of the photosensitizer is in red. Here in parental cells, and here in resistant cells. And when we quantify the fluorescence intensity by spectrophotometry, we saw about double the amount of fluorescence of the photosensitizer in parental cells in blue, comparing with resistant cells in red. And a similar result was found by flow cytometry. These are controls without the drug, and these are cells with the drug. Resistant cells in red and parental cells in blue. In a cross-resistance analysis by MTT, we saw that cells resistant to PDT with methyl ALA in red were resistant also to PDT with ALA. Survival mechanisms based on uptake and or metabolism of methyl ALA and ALA, among others, will contribute to cell survival after treatment. But when cells resistant to PDT with methyl ALA in red were treated with PDT with a drug called verteporfin, they were relatively sensitive to PDT, suggesting that a combination of different drugs will increase the results of the treatment. But we don't rule out that other mechanisms, in addition to those related specifically to methyl ALA and ALA, could be involved, such as an increase in activations of reactive oxygen species, induction of many stress response genes, and an increased repair of induced damage to proteins, membranes, and to DNA. Interestingly, resistant cells not only accumulate less photosensitizer, but they are more proliferative in three-dimensional cultures, as we can see in the quantification of the number of cells per spheroid, here in resistant cells in red, and here in parental cells in blue. And here is spheroids of parental cells, here is spheroids of resistant cells. Parental and resistant population have equal clonogenic growth in 2D cultures. Furthermore, resistant cells were more tumorigenic than parental cells when they were injected subcutaneously in immunodeficient mice, both when 2 injections were made per mouse and when a single injection was made per mouse. Proliferation and tumorigenic molecular weights will be positively selected or induced by the treatment. And when the tumors were analyzed histologically, we saw that resistant cells form tumors with large and diffuse areas of necrosis and small blood vessels that was not found in the parental sample. It could indicate that resistant population have a higher proliferative rate in vivo. And when we measure the messenger RNA levels of different growth factor receptors, EGFR, FGFR, and PDGFR, we saw that resistant cells have an overexpression of these. Growth factor receptors will contribute to cell survival after treatment proliferations, angiogenesis, and tumor genesis. So our humble conclusions of our initial characterization is that resistant populations accumulate less photosensitizer, form spheroids of higher number of cells, had higher tumorigenic capacity, and express higher messenger RNA levels of different growth factor receptors than parental cells. So the next step of our work is to study the role of glioblastoma stem-like cells in photodynamic therapy resistance, the role of the growth factor receptors in the survival of glioblastoma cells to PDT, the migration mechanisms of the PDT-resistant glioblastoma cells, the possible advantages and disadvantages of methyl ALA comparing with ALA, employing different cell lines and primary cultures before moving on to an animal model. And to study the influence of the tumor microenvironment in the resistance. It is our intention to contribute to improving treatment to prolong the survival of people suffering this disease. Thank you to Guido, Torres, Velázquez, Nigra, Casas, Urbina, Bellini, and thank you to Thanks Scholar Conferences.